Hi everyone, on this video I am going to discuss about the Lac Operon. What is Lac Operon and how it is working and why it is working there. Let's go to the video. What is Lac Operon? You have to understand it is a gene regulating system. Why our genes are regulated? You have to understand that there are a lot of genes are there in our body and every gene will code for some particular protein. And you have to understand that not every proteins are needed in our body every time. Our body needs particular protein only on certain times are there. So what our body will do if we don't need a protein? We will say like our body will turn off that gene at that time, and whenever we need that protein, the body will turn on that gene. So how this body turning off and turning on the gene? That is explained by the concept of Lac Operon that is put forward by two scientists, that is known as Jacob and Monod. Let's go and see the details of the Lac Operon to study how this switch on and switch off is done there. Before I am going to the topic proper, let me tell what is this lac in this Lac Operon indicate. You have to understand this thing first because this is for lactose. This lac is for what? Lactose there. So you know lactose is a sugar, and this sugar is available from the milk actually, and this Lac Operon that is working in a bacteria for the digestion of that lactose there. So let's see how this digestion of that lactose is happening in that bacteria. For that purpose I am telling like the bacteria needs three different kinds of enzymes are there. Lactose digestion, bacteria needs how many enzymes? Three enzymes are needed. Which are the three enzymes? You can see the first enzyme that is known as beta galactosidase. You know galactose is the sugar that is present in the lactose, and for separating the galactose from that lactose we need the beta galactosidase enzyme. So I am telling like this beta galactosidase enzyme is the digesting enzyme. It is a digesting enzyme for lactose. Are you understanding? Okay, so the second enzyme, what is the second enzyme? This is known as permease. So what is the function of the permease? You have to understand that the permease allows the lactose to get into lactose to enter the bacterial cell. So this is the function of the permease. Actually I am telling in another ways that permease enzyme will increase the permeability of the lactose to the bacterial cell. That will be more easy to understand. So initially you have to understand the bacterial cell is not permeable for the lactose. So when the permease is there, the permease will increase the permeability so that lactose will enter there. And the third enzyme the bacteria needs is that is known as transacetylase, and that is for the energy purpose. You know every reaction needs energy. So here there is some kind of transacetylation that is for the energy purpose is served there. So we have three enzymes needed. One is for that digestion, another one for that intake, that is for the increasing the permeability, and third one for the energy purpose, that is beta galactosidase, transacetylase and the permease. Three enzymes are needed there. And how these three enzymes are needed? Or you tell me that when the bacteria needs these three enzymes, you have to understand these three enzymes are needed only in the presence of lactose, only in the presence of lactose. You have to understand if the lactose is absent, there, do the bacteria need this enzyme? You have to understand if the lactose is absent, the bacteria do not need this enzyme. Why? Because to save the energy. Lactose is absent, so we do not need the lactose digesting enzymes there. So just see that what happens if the lactose is absent there first. Okay, so you can see this is a figure from your NCERT textbook. You must be very familiar. So how this will be working, you have to understand if the lactose is absent there, what will happen? Before you can see this was the gene for that three enzymes was there, and just before that starting of this gene, you can see there is an O region that is for an operator region. You can see this is an operator region. The operator is the one which is allowing the transcription of this ZYA regions there. So what will happen if the lactose is absent? You can see if the lactose is absent, the I gene just prior to the operator, there is an I gene is there. The I gene will be transcribed and it will be producing a repressor mRNA. You can see that it will be producing a repressor mRNA, and this repressor mRNA will produce a repressor protein. This is a protein that is known as a repressor protein. So I am writing here is RP. Okay, so when the lactose is absent, the I gene prior to the operator will be transcribed and it will produce a repressor. And what this repressor will do, you can see that the repressor will go to the operator. You can see the repressor will go to the operator, and repressor will block the operator. Are you understanding? The repressor will block the operator. So if the operator is blocked, what will happen? So for that transcription of this ZYA gene, you know that will be, there is the RNA polymerase enzyme will come to transcribe this ZYA, and this RNA polymerase will be running through this promoter, and when it is reaching that operator region, what that RNA polymerase seen, the operator is being blocked by some other protein, that is a repressor. So can the RNA polymerase go through this repressor to reach here? You have to understand that RNA polymerase will never reach here. So what will happen? The transcription of this ZYA gene will not occur. Okay, so you have to understand, I will be explaining once again this thing. You have to understand that thing. In the absence of lactose, our E. coli, our bacteria do not need these enzymes. So at that time, there will be an repressor mRNA will be produced from this I gene, that will produce a repressor protein that will come and bind that operator, or actually that will block the operator. So when the enzyme for transcription that will be coming here, the enzyme will be blocked here. So the enzyme cannot reach here, and this area will not be transcribed. Here. So what will happen if there is that lactose is present? You have to understand that thing. The lactose will act as an inducer. You have to understand the lactose will act as an inducer here. So in this condition also, you have to understand, this is very, very important. In this condition also, the I gene will be transcribed. I gene will be producing a repressor mRNA, and the repressor mRNA will produce a repressor protein. That is happening there. But what happens? You have to understand. You can see that inducer. You can see this inducer, and who is the inducer? You have to understand, inducer is the lactose. Lactose is acting as the inducer, and this inducer will come and it will bind on the repressor. You can see the inducer will bind on the repressor. So when the inducer is binding on the repressor, what will happen to the repressor there? The repressor will be getting inactive. You have to understand, the repressor will be getting inactive. So if there is no repressor, what is happening? You can see that if there is no repressor, the operator region will remain active itself. The operator region will be remain active. So on the above picture, what is happening to the operator? The operator was getting blocked. So if there is no repressor, that operator will become active. So at that time, if that enzyme for the transcription is coming, that will be coming here. You can see the operator is active. So what will happen? There will be transcription of this ZYA regions will be occurring. The transcription of ZYA region is occurring, so that it will produce a Lac mRNA. The Lac mRNA will contain the details from the Z region, from the Y region, from the A region. From the Lac mRNA, it will produce the beta galactosidase, the permease, and transacetylase. So this is the thing is very simple. If the lactose is present, the lactose will act as that inducer, and it will block the repressor, so that transcription will be occurring. So let us summarize what we have learned here. So the Lac Operon is for what? Lac Operon is for lactose digestion. Let me write, it is for what? Lactose digestion in bacteria. You have to understand, this is only for bacteria, not for that eukaryotic organism, only in the prokaryotic organism. So there are two conditions are there. Which are the two conditions? One condition, the lactose absent. Okay, so other condition, the lactose present. Okay, so tell me that in which condition that bacteria have to produce the enzyme for the lactose digestion. You have to understand that bacteria have to produce the enzymes only in this condition when the lactose is present, and if the lactose is absent, bacteria do not have to do this thing. So when the lactose is present, what the bacteria will be doing? The lactose act as inducer, and what this inducer will do? Inducer will block repressor. It will block the repressor. Okay, it will block the repressor. So if the repressor is blocked, what will happen? That operator region will be free. Operator is free. So I hope you understand this thing. So when the lactose is absent, you have to understand, when the lactose is absent, what will be occurring there? There is a repressor will be active. Repressor will be active, and this active repressor, what it will be doing? It will block the operator. And there is no mRNA production, no Lac mRNA production there. production. So that's all about that Lac Operon, and you have to understand this figure in detail. And one thing that figure is not telling is that I'm telling like the inducer that is mentioned in this figure, that is actually lactose, that is not mentioned here. And you have to know this is a very simple model of regulation of that gene control. And I'll be teaching entire genetics in that DOPA app in future. So download the DOPA app from that Google Play Store, and you will be getting everything related to genetics from my own words there. For more videos like this, don't forget to subscribe my channel and click the bell button to get every information which I am posting for you. Thank you. Bye bye.